Thank you. It's a pleasure to be here. I'm a postdoc at the Baker Heart and Diabetes Research Institute in Australia, and I'm going to be talking about some work I did in my PhD at the University of Melbourne, developing an R package called NetRep, which you can use to test preservation of network module topology. So the analysis workflow that this fits into is one where you're doing network inference, you're learning the edges from some sort of data set, and then within that data set, that network, you've identified some subnetwork that's interesting, either because it's topologically distinct or it's associated with some sort of trait or disease of interest. And then so the question that NetRep tries to answer is, well, if I take this subnetwork, I go into another data set, is the topology of this subnetwork reproducible in this other data set if I build the network there with either the same or another network algorithm? So this is really important if you want to determine whether your module is reproducible, but it's also useful if you want to start asking questions about is your module present in different types of data sets, for example, different tissues or cell lines than the ones that you've measured, different spatial locations, or over time. So you need 4 different things to use NetRep. You need the data matrix for each of your data sets that you're interested in looking at. So in this example, I'm going to show you gene co-expression networks, although this is a more general approach. You need a matrix of the correlation structure between your variables and a matrix of the network edge weights. And of course, you also need some sort of labeling of nodes and what clusters or modules they fall into in your discovery data set, and so that NetRep can test that in the other networks. So then what NetRep does is it calculates a bunch of topological features of the network in each data set to ask the question, if I look at this network module in the other data set, is the topology more similar than we'd expect by chance, compared to modules of the same size randomly selected in the test data set? And does the module remain topologically distinct? Does the data remain coherent in that second data set? So for those of you familiar with WGCNA, weighted gene co-expression network analysis, we use the same suite of statistics, but rather than using heuristics to assess significance of all of these different module preservation statistics, we do full permutation testing. So our package is much, much faster. That allows us to do full permutation testing to get accurate and unbiased p-values. And this is really important when you want to start correcting for multiple testing. So when you start asking questions like, well, if I have gene expression data from multiple different tissues, and I look at all of the co-expression networks that are in these tissues and start comparing between the tissues, which modules are significantly preserved, which ones are not. So I won't go into those details. So in terms of the package, obviously it's very fast. We've developed it to be very scalable, so you can run it multiple nodes, many different cores, many data sets, so it'll try to keep only one data set in memory at a time. It generates those nice plots of network module topology. So if you were on Twitter before, my personal bugbear is network hairballs. I like heat maps, so this does heat maps. So that's what the package does. If you're interested in it, we have a couple different papers. The main paper for this package, some of the key points are that we show that in real-world data, the null distributions of these statistics are not normal, so you do require full permutation testing, and that sort of explains why if you do z-scores, as in the WGCNA package, you get astronomically high p-value, or astronomically small p-values, sorry. In that paper, we do a pairwise analysis of 4 mouse transcriptomes with 165 gene co-expression networks, and we also look at how these statistics generalize to networks with sparse networks, directed networks, and sparse compositional data, because we wanted to see how general this was. So we also look at gut microbiome networks constructed from 16S data in the Human Microbiome Project. So I'd just like to acknowledge my supervisor, Mike Inoue, as well as other people in the pathogenomics group for helping with the microbiome analysis. I'm at poster A326. You can install the package from CRAN. We have GitHub. Come find me later if you want to look at biological examples of how this works in practice. Thank you.